Today on the Perception and Action podcast, a look at some recent motor learning neuroscience articles. Can we achieve gray matter plasticity through motor learning? How are motor synergies reflected in brain activity? And what are the neural mechanisms underlying the benefits of self-controlled feedback? So it's time for a call to action. Hi everyone, this is Rob Gray from Arizona State University and PerceptionAction.com. Welcome to the Perception Action Podcast, where I discuss how psychological research can be applied to improving performance, accelerating skill acquisition, and designing technologies. In today's episode, I want to look at some recent research investigating the neural mechanisms underlying motor learning and skill acquisition. What exactly is going on in your brain when you learn a new skill? The first paper I want to look at, published by McCrory and colleagues earlier this year, examined gray matter plasticity. For those that may not know, gray matter, named for its pinkish-gray color, is home to neural cell bodies, axon terminals, and dendrites, as well as the nerve synapses within the brain. This brain tissue is abundant in the cerebellum, cerebrum, and brainstem. It also forms a butterfly-shaped portion of the central spinal cord. White matter, on the other hand, is composed of bundles of axons. These axons are coated with myelin, a mixture of proteins and lipids that helps conduct nerve signals and protect the axons. White matter's job is to conduct, process, and send nerve signals up and down the spinal cord. Gray matter density has been shown to be a measure related to important health outcomes. Its loss is associated with cognitive declines in aging, dementia, Alzheimer's, and Parkinson's disease. Increasing or maintaining gray matter density throughout adulthood and older ages seems essential to prevent these adverse outcomes. Previous research has shown that slowing gray matter density loss can be achieved through exercise by developing a high level of aerobic fitness. To date, the benefits of exercise on gray matter density have primarily been thought to be related to cardiovascular capacity. However, in this current paper, McCrory and colleagues argue that there's more to it than just this. Specifically, they propose that activities which require motor learning and provide new movement possibilities are more efficient at inducing plastic changes in the gray matter density, as opposed to just repetitive physical exercises. In other words, they are proposing a theoretical link between a motor repertoire enhancement and gains in gray matter density and plasticity. Within this paper, the authors define motor repertoire as the number of movement possibilities and motor skills that can be performed by a person. For example, a martial arts athlete has more extensive motor repertoire than a sedentary person, due to the movements learned and performed in their sport. Thus, learning a new motor skill, as well as experiencing and being able to perform different movement combinations, expands one's motor repertoire. I also really like their definition of plasticity in this paper. Quote, a mismatch between the demand of the environment and the current capacity of the system, in this case learning a new motor task, represents the starting point for the modifications in brain structure. Furthermore, this mismatch must be sufficiently sustained through time to induce the plastic outcomes, and also be challenging enough according to the individual flexibility and capacity of each subject. End quote. So let's look at this proposal in a little bit more detail and consider evidence that might support it. Learning a novel motor task demands considerable brain activation due to new processing of input and output information regarding attention, sensation, perception, decision-making, memory, and so on. Therefore, performing a new movement stimulates distinct brain areas, thus inducing positive plastic changes in the corresponding region. The consolidation of these new motor tasks expands the individual's motor repertoire and promotes the plasticity in the gray matter density. As a possible mechanism responsible for this functional plasticity, the authors propose that improvements in performance with practice are due to reorganization of cortical synapses in the motor cortex. One piece of evidence the authors put forth to support their idea is a 2014 study by Schlafly and colleagues, which compared the brain morphology of endurance athletes and martial arts athletes with a non-athlete control group. High-resolution structural imaging of the brain revealed higher gray matter volumes in the supplementary motor area and the dorsal premotor cortex in both athlete groups as compared to the control group. Another study discussed was a 2008 paper by Draymire and colleagues. In this study, it was shown that learning a 3-ball cascade juggling skill altered gray matter in the occipital temporal cortex as early as 7 days after training. After 5 weeks of training, significant increases in gray matter density were also found in the cingulate cortex, medial temporal gyrus, superior frontal gyrus, and inferior parietal lobe. These findings suggest that learning of a new task is critical for the brain to change its structure, inducing plastic modifications very early after training onset. Additionally, it's important to note in this case that juggling training did not involve high cardiovascular demands. Finally, in a 2004 study by Drudzinski and colleagues, it was shown that 3 months of juggling could increase gray matter density in the mid-temporal area and the parietal lobe. Interestingly, after 3 months without practicing, those changes in gray matter density were not sustained, as well as the performance on the juggling skill got worse. Thus, this data suggests a transient change in brain structure due to a novel motor stimulus. The demand of learning a new motor skill causes the initial mismatch that promotes the gray matter gains in the given area, whereas the lack of practice promotes another mismatch, with a downwards adaptation, reverting the gains previously acquired with practice. The second study I want to look at is one published by Zandvoort and colleagues earlier this year in the journal Neuroimage. This one looked at the brain activity associated with the development of muscle or motor synergies, which of course is something I've talked about several times on the podcast now. Check out episode 135, for example. The idea here is that one of the main ways we take advantage of or control the multiple degrees of freedom in our movement system is by developing task-specific combinations of muscle activity, which we call muscle synergies. In this current study, the authors examined the potential link between motor cortex oscillations and muscle synergies at specific frequency bands. When performing a motor action, we obviously get neural activity in large populations of neurons. Research has shown that in some cases this activity is phase-locked. In other words, it increases and decreases at the same rate. Another term that you will commonly see used to describe this effect is entrainment, which basically means the synchronization of brain activity over a specific frequency range. Entrainment, or phase locking of neural populations, facilitates information transfer between them and is apparent in the motor system at various levels. It also seems to occur at different frequencies. For example, the beta frequency band, 15 to 30 Hz, is believed to reflect the entrainment of spinal motor neurons and the cortical motor nuclei. The 30 to 60 Hz frequency range has been labeled the Piper rhythm, and it seems to be involved in dynamical tasks like locomotion, balance, and manual control. This rhythm seems to also serve for correcting errors. In the study by Zandvoort and colleagues, the authors investigated functional cortico-synergy connectivity and its plasticity in short-term postural training, that is, learning to balance. It was hypothesized that Piper rhythm coherence between the motor cortex and the muscle synergies to be modulated as a function of practice. The study involved 15 participants learning to balance on one leg on a robot-controlled balance board, which introduced rotational perturbations. Training involved practicing on a standard wobble board for 30 minutes. Pre- and post-tests of 10 trials each using the robot board were conducted. EEG, EMG, and kinematics data were collected. Of course, the main measures of interest were the time locking between the different signals. To quantify balance performance, the time to recovery after a robot perturbation was used. What was found? The activity of the 9 muscles recorded from could be decomposed into 3 synergies. For example, Synergy 1 comprised temporal activation patterns of the gastroc, soleus, and rectus femoris. Connectivity estimates revealed significant coherence between muscle activities in these motor synergies and the activity in the left paracentral lobe around the Piper rhythm. Finally, cortico-synergy coherence around the Piper rhythm decreased as a function of postural training, which did result in faster times to respond to perturbations. The authors propose that this decrease occurs because less coherent, smaller populations become involved in finer level of control with learning. So what does all this mean? To sum up, in the study it was found that there is a Piper rhythm coherence between the motor cortex and synergistic muscle groups of the contralateral leg. In other words, the cortical spinal tract conveys the Piper rhythm from the motor cortex down to groups of muscles simultaneously rather than to individual muscles. Cortico-synergy commands may be considered higher-order regulations to meet the specific postural demands. So in other words, controlling action through the development of coordinated structures rather than programming the value of each individual muscle. The last study I want to look at in this episode, published by Kim and colleagues this year in Human Movement Science, looked at the neural mechanisms underlying the benefits of self-controlled feedback, which I first discussed way back in episode 33. As previously discussed, There are two basic explanations put forth to explain the benefits of self-controlled feedback. First is the idea that it has cognitive benefits. That is, the performer is actually using the information in the feedback to correct errors, and they're doing it better because they're getting it when they need it most. The second explanation is a motivational one. That is, the mere choice of when to get feedback makes the performer more motivated to perform well, regardless of the information carried in the feedback. For example, see the optimal theory of Wolff and Luftwait. The goal of the present study was to further examine this issue by looking at brain activity using the typical self-controlled learning paradigm. Specifically, this study examined event-related potentials, or ERPs, in brain activity. ERPs are changes in brain activity to a specific event. The most well-known example is called the P300. This is a positive increase in brain activity that occurs roughly 300 milliseconds after an unexpected or oddball stimulus is presented, and it's thought to be involved in decision-making. In the present study, the authors predicted that the self-control group would show superiority at processing task stimuli, response errors, and feedback compared to the yoked group, and this would all be reflected in larger P300 amplitudes. In the study, 42 high school students learned a 6-digit key pressing task, in which they had to press a series of keys while also trying to maintain a total goal movement time of 2.5 seconds. Following the standard paradigm, participants were split into two groups, one that could receive feedback whenever they chose, and a yoked group whose schedule of feedback was matched to one of the participants in the choice group. The feedback contained three types of information. A, whether the keys were pressed correctly or incorrectly. B, the participant's response time. And C, how much they would have to adjust their movement time for the next trial. For example, slightly more slowly or slightly faster. The acquisition phase consisted of 4 blocks of 50 trials each, and there was a retention test after 24 hours. 2 transfer phases followed the retention test. In the first transfer phase, both groups were required to respond to the same stimuli, but with a novel movement time of 3.3 seconds. In the second transfer phase, the participants were asked to respond to a new pattern of 6 digits, but with the same original 2.5 second movement time. What was found? Analysis of the key pressing errors during the 2 transfer tests revealed the typical effect. The yoked group made more key pressing errors than the self-control group. But what about the ERP results? Event-related potential analyses revealed that the self-control group exhibited larger post-stimulus and post-feedback P300 amplitudes than the yoked group in the frontal regions of the brain. These results suggest that self-controlled feedback may allow the learner to more effectively process the task stimuli and feedback information, and also contributes to enhancing the learner's motivation and attachment to the task being practiced. So in other words, this study provides support for both proposed mechanisms. Okay, that's it for today's episode. Remember, you can contact me at robgray@asu.edu or follow me on Twitter at ShakyWeights. To find out more about the podcast, please check out perceptionaction.com. Finally, to support the podcast and receive bonus materials, including an extra monthly episode and written transcripts, please head over to patreon.com/perceptionaction. This is Rob Gray from ASU. Cheers for now, and keep them coupled.